Let's go ahead and get started. As usual on Wednesdays, I will probably plug the biology department seminar. This week is Dr. Tim Nice, I think that's how you pronounce it, from OHSU, who will be presenting on enteric viral infection. Now, what the heck is enteric viral infection? I had no idea what this was. So I went and took a quick look. It's really about viruses in the microbiome in your gut and how those then interact with the microbiome and then interact with us in terms of making us sick. So that should be a lot of fun. Those are always from noon to 1, and I'm pretty sure that this term we're in Science Building 1, room 107. So that's the big auditorium in Science Building 1 on the ground floor. If you get there a little bit late, please come in the back door. So again, highly recommend checking these out. No, there won't be anything on various exams as far as this is concerned. So now, let's get started with Lecture 2, virology, all of these fun things again up here on the screen. Today we're mostly going to be talking about virus counting and how you go about counting something that generally you can't see. But first we'll talk a little bit about the history of virology, particularly in terms of vaccines, because that's some of the very earliest. In fact, vaccines were discovered before we even knew about viruses. It was a way to deal with disease, and smallpox was the very first of those. And when we talk about pox viruses later on in the term, we'll talk a little bit more about these things. Pathogens, we talked about definitions last time. The first viruses were found, again, getting back to this vaccine thing, because they were pathogens, but they were really little pathogens. And that's basically the way that all of these viruses were discovered. And we'll talk a little bit about that. But mostly today we're going to talk about how you go about detecting viruses. And particularly important here is detecting infectious viruses. And so you can detect virions as much as you like. But if you're really interested in the progression of the whole virus life cycle, then you have to know something about infectious viruses. And that has a lot to do with these plaque assays. And these are just a couple of plaque assays that I have actually done in our lab, and we'll talk a lot more about how these are done. Very classic technique. Any of you who have listened to This Week in Virology know that the This Week in Virology crew are big fans of plaque assays. I am as well. The other important concepts that we'll talk about today are the so-called one-step growth curve. This is a way for studying virus replication. And again, it's that whole virus, not just the virion here. In order to study a one-step growth curve, the whole idea here is that it's very, very, very difficult, although now slowly becoming possible, to study one particular virion infecting one particular host. Usually you've got to look at bulk. And so the idea of the one-step growth curve is to see what's happening in bulk, but back calculate that to the one virion going through that one virus life cycle. And so the whole idea here is synchronizing that you have a completely infected set of hosts. And in order to do that, we need to do some math. Math, scary stuff. So everybody clear on the Poisson distribution? No? Okay, we'll spend a little time talking about it. I figured as much. That's usually the case. But first, let's roll back the clock a little bit. And interestingly enough, only a little over 100 years ago, the very first viruses were described as such. And at that point, communication wasn't great. So basically, independently by a Russian researcher and a Dutch researcher, they found that a disease that infected plants was really, really small, and they couldn't make any of it, i.e. they couldn't get it to grow. This is exactly the same time that Pasteur and Koch were working on microbes and spontaneous generation, and you didn't have any spontaneous arisal of microbes in the absence of being connected to air, etc. But one of the things that they found, again with Pasteur and Koch, if you filtered your bacterial culture through a very fine filter, you got rid of anything that could grow, and you couldn't cause disease until these, again, Dutch and Russian researchers came along and said, hey, we've got these filters that get rid of all life, but there's still something that causes disease that can come through them. That we now know as tobacco mosaic virus, and this is a transmission electron micrograph of those. We saw these before. These are really classic helically symmetrical virions, and we'll talk a little bit more about tobacco mosaic virus, or TMV, a little bit later on. Ivanovsky didn't really follow up on this study, whereas Bierjink really did. And I shall try and link to his original paper here. How many people read German? Okay. How many read English? Good. Okay, I'll trust the translation of this. It's really an amazing paper. In 1898, he came up with virus. He had some really good ideas about what virions were. I was extremely impressed when I finally got around to reading this paper. But basically, the message was really small, didn't grow in any kind of conditions, and was, in fact, the disease-causing particle, and he even talked about it as a particle, was really pretty resistant to all kinds of things. And it turns out that tobacco mosaic virus is so resistant that if you just go through cigarette butts, I've been told, you can actually find tobacco mosaic virus in a lot of these. So interesting kind of aside there. Very soon thereafter, actually in the same year, the foot and mouth disease causing agent was also found to be really small, and they just couldn't grow these things, try as they might. Now, any of you who do microbiology know we can't cultivate those microbes either. But this was the first indication that they did actually require some kind of host in order to replicate. And again, this was just a disease-causing agent, and it was really small, so it was originally called a fluid virus, this fluvium. Contagium, of course, infectious. Vividum is this, maybe virile, potentially alive, and then fluvium, fluid. So this is originally what they called it, the now ultrafilter were those filters that held up all bacteria, virus, which now we're just calling a virus. And of course, we shouldn't be calling this a virus, we'd be calling it a virion. Good, you guys are quick. The second sort of, I think, major discovery in terms of discovery of viruses was also independently done by two different researchers, now one in England and one at that point in French-speaking Canada and Quebec, Twort and Derel. Derel is very famous for working on these viruses of bacteria. So bacteriophage, phage just stands for eating, so whatever eats bacteria, later found to be these sort of disease-causing agents as well, also very small in terms of filtration. Félix Derel is really sort of thought to be the father of the bacteriophage and thinking about particular things in terms of phage therapy. And so there are a number of actually Derel institutes that we'll talk about a little bit later on here. So one of the immediate thoughts, particularly having to do with this filtration, so you filter whatever you have, this disease-causing agent, whatever comes through, you can't grow it, so it's not alive, but that's the virion, which is potentially not alive. So there's been many, many debates over the years on whether viruses are alive. And this is what I threw up at the end, I think, of last lecture. Are viruses alive? There's no good answer to that question. In fact, I've asked it on problem sets before in the past, and I accept both answers, yes and no, because it really depends on what? Exactly, your definition of what's alive. And so my standard answer to this, which is complete obfuscation, is viruses are part of life, which means I avoid the whole problem at that same time. But a couple of the issues that people had with viruses being alive or not. One of them, and this was in fact from the Nobel laureate Wendell Stanley, who crystallized tobacco mosaic virus and said, you can't crystallize life. there's no way that you can crystallize life. But you can crystallize virions. However, the sort of standard definition of life that NASA uses when they're going to other planets and moons and things and trying to detect life is something which is evolving. And viruses clearly evolve. So now we've got these two different things here. Eckhard Wimmer, who's done a lot of work on polio, is pretty famous for publishing the molecular formula for polio. So C332,652, H492,388, etc. So how the heck can you put a chemical formula on something which is alive? Again, it's just the virion, though, in this case. And there's a whole discussion about this here on this website. And then, viruses clearly reproduce. So you can always use, again, the obfuscation that I do. Viruses are part of life. Or have wonderful long-term debates, preferably with some alcohol or whatever your favorite poison is, talking about whether viruses are alive or not. Whether they are alive or not, we've learned huge amounts about how cellular life, that most people usually don't argue about whether it's alive or not, in terms of understanding particularly the very fundamentals of how biology works. So one, and this is, these are just long et ceteras down here at the bottom. So the so-called bacteriophage group, founded by Max Delbruck and Salvador Luria, who was the one who had that really nice definition that I liked for the extracellular elements. They used viruses, and particularly bacterial viruses, to show that DNA was the genetic material, that recombination happened, that we had promoters, et cetera, et cetera, et cetera, et cetera, et cetera. In terms of eukaryotic molecular biology, particularly tumor viruses, and we'll talk about tumor viruses later on in the term, they were used to understand replication origins. And I think even in the molecular biology course last term, we talked about the helicase that's important in replication. And in, I think, the textbook, it said large T. That's actually a viral helicase. Has nothing to do with normal cellular replication. But when people were originally studying replication, that's what they found. And so the large T antigen from SV40, which we'll talk about a lot more later on. Cancer, the reason we know about oncogenes is due to the study of viruses and what are called the transforming viruses. We'll talk about that a little bit later on. And then the whole retroviruses, understanding reverse transcription, which we thought was just a cellular thing, now we know is a viral thing. And in fact, This Week in Virology, I think 2 weeks ago, talked about how insects protect themselves from viruses through this reverse transcription process. There are all kinds of cool things you can do with viruses. Again, this could go longer and longer and longer here, but I specifically wanted to talk about phage therapy, bacteriophage therapy, because this was really introduced by Felix Derel soon after the discovery of these bacterial viruses, because one of the very first bacterial viruses that he found was a bacterial virus that killed cholera, which is a very convenient thing to be able to do, particularly in the late 1800s when there was, beginning of the 1900s, when there's large amounts of bacterial disease. And so what Felix Derel did was he isolated the bacterial viruses, grew large amounts of them, and then actually used them to treat disease, which was great, worked very well. And then antibiotics came along, and so we just completely forgot about phage therapy for the most part. Now, of course, we have all these antibiotic-resistant bacteria. So how do you deal with antibiotic-resistant bacteria? You find a virus which can infect those antibiotic resistance bacteria and use those to treat that particular bacterial disease. There's a group at Evergreen State College, just up I-5 here in Washington, Betty Cutter's group, that's done a lot of work on this, and there's more and more discussion of trying to use these bacterial viruses to treat bacterial diseases. This is a nice iBiology. You guys all know iBiology? iBiology is a wonderful set of videos about sort of fundamental aspects of biology. This particular one is by Paul Turner, who's a colleague of mine from Yale. Does a really nice job talking about viral ecology, the beginning, his first two talks, and the third one is talking about this whole idea of phage therapy. But it's not just phage therapy, as I talked about last time, one of my definition of viruses are bags of genes. And so they like to move genes around. If you have a particular gene that you want to move around, say get into a cell which is lacking a particular gene, or a cell which you want to get rid of a particular gene, viral vectors, which is basically taking a virus and tweaking it in some way, is really critical for gene therapy. Probably 80% of gene therapy, and some of you may have heard there's a new gene therapy for treating some eye diseases, which was just approved by the FDA in the US, completely based on viruses and the ability of viruses to move this genetic material around. Some more far-fetched things have to do with nanotechnology. People are actually using some of these bacterial viruses, but also things that are a lot like that tobacco mosaic virus, to make batteries, because they're really, really small and very uniform, the virions are, and so this being used as templates. There's a whole series of things about cancer therapy. Again, I have a link here. And then finally, as far as phage therapy is concerned, I posted a nice return of the phage article on D2L. You can take a look at this. I'm not going to be testing you specifically on it, but if you're interested in phage therapy, these are some of the websites, and this is kind of a nice reading to look at there. So cool stuff that you can do with viruses. So any questions? I didn't print out my list yet to ask any one person, but I will do that in terms of what we've talked about here so far. Do you have a question, or you're glad I don't have the list yet? Okay, I promise I will do that. Just need to get around to doing it. Okay, so let's shift gears a little bit and then talk about how you go about counting things which are so small that they go through filters, and they're so small they go through filters means you actually can't visualize them directly. And even if you were able to visualize them, it would just be virions that you would be seeing. Now, it's kind of hard to tell sometimes when you've got these infected cells. There are ways to study infected cells as well. But if you're interested in an infectious virus, there are a few ways that you can do it. You can't actually directly image them. And to do that, you need an electron microscope, which, how many people have done electron microscopy? I know that guy in the back has. So you know that it's really pretty complicated, expensive, really big instruments, and counting is a real pain. And I can ask you, some of you, more of you have done an electron microscopy. But actually, doing the counting of the individual virions is very difficult. Plus, you're counting virions, so you have no idea if they're infectious or not. So if you're interested in infection, in particular if you're interested in infection from a pathogenesis point of view, how are they making whatever it is sick that's being infected, then animal work is one way. I should say whole organism work as well. So you can also do this with plants. Basically, take certain amounts of your virion, your virus preparation, and feed them to mice, pigs, hopefully not humans, and see how many or how much do you need to get an actual infection. And that is doable, and it is done in some cases, particularly if you're very interested in pathogenesis. Some of you may have heard about some of the flu experiments. A lot of flu experiments use ferrets. It turns out that ferrets and the way that influenza infects ferrets is very similar to the way influenza infects humans, but really expensive and very hard work to do, and just a relatively small number of animals that you can treat. And if you're really interested in how many animals get sick, you have to have animals that don't get sick, animals that do get sick, etc. So statistics are really kind of tough there. So the way that the vast majority of virologists do these infectivity experiments is something called a plaque assay. And that's shown here in the diagram. Basically, you have your suspension of whatever it is that you're interested in seeing, how many infectious virus particles, infectious virions, do I have in this small amount of liquid. Now, you remember last time I showed you that tube, 10 mils of 10 to the 11th plaque-forming units in it. So you can get a huge number of infectious virus particles in very small amounts of liquid. Same thing again is true with seawater. Anywhere between 1 million and 10 million particles, not all of them are going to be infectious, that are present in any one of these particular samples. So you do tenfold dilutions. This case is just shown with 0.5 mils of whatever you start with. 4.5 mils, that's a 10 to the 1 dilution, 10 to the 2, 10 to the 3. Just take 0.5 out of each of these. And eventually, when you get to this final point right here, this would be a dilution of 10 to the 8th. Often you don't know how many that you're looking at, so you look at various different dilutions here. Then what you do is take this dilution, mix it together with a vast excess of host cells. So this only works if you have a cell culture system. So if you happen to be looking at a virus that infects an organism that you don't have a cell culture system for, you're kind of stuck with the animals or with the plants. But fortunately, this works really well for bacterial viruses and then a number of animal viruses as well. So you mix them with a vast excess of hosts. And then, really important here is you incubate. Now, why are we incubating here?
Growing bacteria.
So you're letting the cells grow. And so this would be bacteria or also, in the case of any of the mammalian cell cultures as well. Exactly. So that's allowing, particularly important, is the cells aren't infected to grow. But then those that are infected will produce more infectious virus particles, which will produce more infectious virus particles around those, infect the cells around that, and produce even more. And eventually what that will do is give you a plaque. And so that's what each of these little dots, hang on, back up here, are supposed to represent here. This lighter dot is where you have less cell growth, or in some cases cell death. That's where you've had infection originally of one infectious virion, but it's now spread. You can then, from counting all of these plaques on these individual plates, you can now back calculate, given that this was a 10 to the 8th dilution, the average on these plates is 12. So 12 plus 15 plus 10 plus 11 divided by 4. That would give you 12 average plaques in this diluted amount that you had here. And then your dilution, 10 to the 8th, gives you 1.2 times 10 to the 9, which is a pretty typical titer that you would see for some of these. Another way of looking at this is getting back to your comment about the bacteria. So if you're just using a bacteriophage, here's another way of doing it. And important here is this top auger, which is used. The important thing about the top auger is that many bacteria like to move around. And so you want to prevent them from moving around. You also don't want your virus diffusing all over the place. So you want to have some way of slowing that down. And so that's what this top auger is about. And then you have this, you mix together this suspension, you pour it onto an auger plate. This is misspelled in the textbook. And then let them sit, and again you get these plaques. And these are some, as I showed you before, some plaques of Phi-X174, which is a single-stranded DNA virus that we'll talk about later on E. coli. And so literally what you do is you take a Sharpie and count each of these little plaques. In this case, I had 67 plaques. Here I had 81. It ends up being an average of about 75. And in fact, if I remember correctly, this is what I used to get that titer of that tube that I was showing you before. So this must have been, on the order of a 10 to the 8th or 10 to the 9th dilution. You want to have between 10 and 100 plaques to count to get a good statistically significant amount here. What's the difference between this and transformation of cells? So colonies versus plaques. And so what you're thinking about when you're talking about doing colonies, if you had this, let me back up here and talk about this. So if you didn't have any virus here and you just replaced this virus with bacteria or with cells, did this dilution, each of these spots would be where your cells were growing. Right, so those would be your individual colonies. Here, you're mixing with a vast excess of host. So what you have in the background, let's go on to this slide, right here, all of this light brown stuff, that's basically what we like to call too many to count number of colonies that have all grown together. So we'll call this a lawn. So this is the lawn of hosts. These are all the uninfected cells which are there. And it's the infected cells which are going to produce each of these spots, which again will then cause virus to be produced and infect the cells around that, etc. That make more sense?
Yeah.
Good.
Okay, just to summarize, the plaques are the infected cells that we're looking at.
Okay, so the plaques, the question here is the plaques the infected cells. The plaques are due to the fact that these cells have been infected, and the reason that you can see through them, there's a plaque, and basically it's just a hole, and this is why Felix Serral called them bacteriophage in the first place, because they were eating a hole in the bacteria, because you couldn't see them. Now, these were actually cells that had died. You can also see cells that have other kinds of phenotypes as well.
And that's why it's the
Right, and so that's why it's the plaque-forming unit. Each of these individual spots represents a plaque. And if you were to take that particular plaque, just that plaque, and then do one of these plaque assays with it, you would find that there's about 1 million infectious particles in each of those plaques because they've got all of these chances to replicate and make more of them. Okay, so also have an example here of mammalian cells. This is one that I did, so I'm particularly proud of it here. Instead of having regular-sized Petri plates, we have 6-well plates. It's a little easier to work with these number of cells. And also dilution series, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5, and 10 to the minus 6. And this is a no virus control. So here, the cells you actually can't see. They aren't brown. You have to stain the cells. That's crystal violet. That's what this blue thing is here. Here we have all of these plaques that have run together. We like to call this TMTC, too many to count. Here, 10 to the minus 3. You got a whole bunch of plaques here. It's kind of tough to count them. Here you start to get a countable number, and here you pretty clearly have 2 plaque-forming units. And I happen to have brought an example of one of these here. So here's our, oh, yeah, the light here is horrible. Sorry about that. But this is a real plaque assay that was actually done by one of my undergraduate students. Again, a no virus control up here, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. And after class, people can come take a look at this later. Yes? Question. I'm just repeating this so that it's on the recording. Do you have trouble with plaques overlapping with each other? And then you count 1, but it's actually really 2. That's one of the reasons you want to get to a dilution in such a way that the plaques are pretty well separated from each other. And you want to do multiples. So in that example that we looked at before, definitely the 5x174, those 2 plates at the same dilution. You also have multiple plates at the same dilution. And this is just one example here that we did for our, and this is a yellow fever virus, by the way, that we're using here. The vaccine strain, not the one that will make you really sick. So these are pretty classic examples. This is done literally all the time, in virology, classic process, and this is just what we looked at already. So plaque assays are great if you happen to have a nice cell culture system and some way of detecting that the cells are dead. There's also something called a fluorescent focus assay, which is where you detect infected cells rather than just seeing that they've died in some way. And so that's another way of doing these processes. Another old school technique for looking at virus production, and an incredibly cheap way of looking at virus production, is something called hemagglutination. And we'll talk about hemagglutination a lot more later when we talk about flu. Everyone heard about H5N1, H1N1, H2N2, etc. What's the H stand for? Hemagglutinin. Why is it called hemagglutinin? Because it does this. So hemagglutination. So it's literally that protein. And what is hemagglutination? All it is clumping red blood cells together, getting red blood cells to bind to each other. So agglutinating red blood cells. In the case of flu, it's binding to sialic acid, which is a extracellular component, sticks out of red blood cells. And basically, you can do the same exact thing here with your dilution series, only now instead of mixing it together with host cells and plating it on a lawn, you mix together the red blood cells with your dilutions of virus. And if there's something in there which will hemagglutinate, i.e. get all of these cells to bind to each other, you'll have a clump, and this clump looks a lot like this down here at the bottom. So this is clumping in such a way that it doesn't go down to the bottom of the tube and make a nice little spot. So this little spot here is the absence of virus. So here's a control where we have no virus whatsoever. All the way across our dilution series, we're just doing dilutions of buffer here, spots. Here, if you have virus at these low dilutions, you have a hemagglutination which has happened. Here are also some hemagglutinations which has happened here. And we're just doing dilutions. So 1 to 4 dilution, 1 to 8 dilution, 1 to 16, 1 to 32, etc., all the way across this 96-well plate. So this is really nice here because we're able to test 8 samples just on a regular 96-well plate here, as opposed to a plaque assay where we're looking at one particular analysis of one kind of virus here. So it's very fast. It's relatively cheap. Main problem is it's not very sensitive. And there are some other issues, particularly if you don't have a virus that can cross-link red blood cells. So here again, I showed you our negative control. Here's our positive control. And then here are 6 different samples. D and E are clearly positive. F and G and C are clearly negative. H is a little harder to tell here. Here's an example of a real hemagglutination assay, which again I did in my lab. This is what it looks like before you have the hemagglutination that takes place. And then after an incubation, I've got our positive control up on the top here. And these are 4 different samples. You can see that we're starting to get this hemagglutination happening. Actually, it's happening over here because we're not seeing these spots. When you do start to see the spots, again, that's the lack of hemagglutination. These spots now over here, but here we've hemagglutinated at these lower dilutions in all of these different samples here. So hemagglutination, great, quick, but relatively low sensitivity, also very cheap in terms of doing these assays. And you can look at a whole bunch of things simultaneously this way. Okay, any questions? Again, where's my list? Yeah, in the back. Okay, so what are my controls? Obviously, I'll paraphrase your question here and correct me if I'm not understanding correctly. So there are really 2 controls. And whenever you do an experiment, you want to have a positive control and a negative control. So the negative control is just buffer. So you have no virus in there whatsoever. And in this particular case, I didn't do a negative control. Don't tell anybody. Then if you have a positive control, that's going to be a known amount of infectious virus or something which is going to hemagglutinate. And so then you can compare that to whatever your experimental things are. So really you want to have 2 kinds of control. So known amount of infectious virus, or in this case something's hemagglutinate, and you'll have your negative control, which is just nothing. It shouldn't be in there at all. Yeah, and this is true not just for plaque assays, but experiments in general. Negative controls, positive controls. People in my lab say if the only thing they've learned is that you need controls, if that's the only thing they've learned, I consider my job done. So there are a few caveats with these assays, and even with hemagglutination. Hemagglutination, as I've been trying to emphasize here, that's just binding red blood cells together. So you actually could have non-infectious viruses, which will still get red blood cells to hemagglutinate. And again, if you're doing electron microscopy, you can see lots of virions. You have no idea if those are infectious at all. And in fact, what people have done is now done these kinds of comparisons. And what you find, in many cases, the number of plaque-forming units, again, PFUs versus CFUs, can sometimes be way, way, way smaller than the number of virions. And in some cases, particularly for animal viruses, up to 1,000-fold lower. So you have huge numbers of virions, most of which are not infectious. So why is that? As we'll see later on, assembling virions is sometimes really challenging. So that's a definite possibility that some of these virions are actually incomplete. They're haven't been made properly. Defective genomes, this is something we're finding that is more and more common, again, particularly in animal viruses. You have a perfectly good virion, but whatever gets packaged inside it, that genome has problems. And then it's not going to be able to undergo full replication, so you're not going to be forming plaques. In some cases, you'll have empty capsids. And in fact, you can often see these in the electron microscope. And then, if you're using a cell which has an active antiviral defense system, sometimes you can have no plaques whatsoever. And this is one of the things when you're trying to set up an assay in the beginning and you don't have a good cell culture assay, you have to be very careful about what cells you're using. You have to, again, it's that positive control that we talked about just a second ago. Make sure you have something which is going to give you these positive controls. So if you have a plaque assay, it's awesome. If you don't, there are other ways of doing things, but they're a lot less useful in terms of thinking about infectious virus particles. So infectious virus particles are great. We'd love to be able to study a whole infection with the one individual virion going into a cell, going through the whole virus life cycle, et cetera. But it's really, really hard to study one virus, I say one, and the one cell, the one virion, one cell process. So usually these are done through what we call one-step growth curves. So the whole idea of a one-step growth curve is that you have a bunch of virus and a bunch of cells all infected at the same time in, relatively speaking, the same amount. And so if you follow all of these cells together, they'll all be producing various components of the virus and eventually virions simultaneously. And so the point of this is to, again, use that average to back understand how that one virion is infecting the one particular cell. Works most of the time. But again, the key to this is having a simultaneous infection and hopefully a synchronized infection so all cells get infected at the same time. So one way of looking at this is a really typical one-step growth curve. Particularly important here, so let's look at this black line. So the black line here is infectious virus. How do you know that this is infectious virus? Plaque assays, exactly. So PFUs. So you start out here with a PFU per cell of 10, and we'll see why that is in just a second. That goes down because you're really interested in infected cells and that process of the infection going on to new virions. So this goes down, and we'll talk about how that happens. Gets to a pretty low level, and after a certain period of time, and this varies from virus to virus, you'll start to see more and more infectious particles, and eventually this will level out. One-step growth curves are almost always separated into two phases, the so-called early phase and late phase. Again, the virologists are incredibly creative in terms of their names here. Early infection is when you've gone to seeing no virus particles. Late infection is when you start seeing virions. And this can vary a little bit here as well. If you think about what you need, in this particular case, this is a DNA virus, so it has a DNA genome inside each of the virions. So the first thing that happens is this genome that's been released. Here, all these infectious virus particles have gone down to 1 per cell, and we can talk about why that is later on. You start to see production of the messenger RNA from the virus. Then you start to see proteins, and in this case, it's the T antigens, but these are just proteins. So you start with RNA, then you start to see protein, then you start to see viral DNA, and then you finally start to see these capsid proteins, and then infectious virus. So all of the interesting stuff from a molecular virology point of view is happening in this part here because you're getting transcription, you're getting translation, you're getting replication, you're getting assembly of virus particles, and then we have the final release of the virus particles here. So how do you do these? Again, it's just looking at a plaque assay over time. Usually it's going to be this dark line, but inside of that you can see all of these other important molecular aspects of replication that are happening. So how do you do one of these one-step growth curves? That's here. You now sort of flip this whole process around. Remember the plaque assay where you use small amounts of virus and a vast excess of host cells? Now you want a real excess of virus relative to the host cells. Why is that? Because you want all of those cells to be infected, and you want them to be all infected at the same time. And that actually gets back to this, which I forgot to mention, high MOI. What the heck is MOI? Well, have you done your reading? Multiplicity of infection, exactly. So when I get my list, I'll be calling you out on this. So what's multiplicity of infection? It's the number of infectious particles per cell. And so in this case, what are we starting out with? An MOI of 10, exactly. So that's 10 times as many infectious particles as we have cells. Once you've mixed these two together, then you do a dilution or a wash step. Why might you want to do this? And no answering in the back, Nacho. Getting rid of excess viruses, exactly. And so here, this reduction right here. Now, why is this important? Because you might then have extra infections that are taking place that would really mess up the rest of your chronological infection process here. So then, again, just do your plaque assays over time, and you'll see something that looks like this. This is your idealized curve. So virus added here, just the black line again is representing infectious viruses. Here's our early part. Here's our late part. And very often, people will talk about this as the latent phase. The latent phase is the absence of any new infectious particles, because you would have had some infectious particles right here at the beginning. And then the assembly and release phase right here. People will also talk about an eclipse phase versus a latent phase. The eclipse phase is when you don't see any infectious virus particles either inside or outside the cell. And in order to see if there's any that are present inside the cell, you actually have to artificially break open the cells to see intracellular viruses. So up to the end of the eclipse phase, this you now have had all of the proteins that you need. There'll be what we call early proteins. Early proteins are important for replicating the nucleic acid of the virus, because you need to make that nucleic acid to put it in the bag. What's the bag? Those are the capsid proteins. You need to make those. And then once you have nucleic acid and the bag, that can then get put together. And at the end of that will be the end of the eclipse period. If there's a difference between an eclipse period and a latent period, that's the accumulation of virions inside the cell before they come out. And so we can look at a couple of examples here of that. These are 3 different examples right here. Actually, 2 different examples. This is just bacterial growth. Adenovirus and Western equine encephalitis virus. Adenovirus, we'll talk about later on, is a naked virus, i.e. it doesn't have an envelope of lipids around the outside. Western equine encephalitis virus is an envelope virus, and that envelope is picked up at the cellular membrane, so literally as infectious virions are being produced. What you see in terms of adenovirus is actually also quite similar to what you see in the case of most bacteriophage. You have this typical drop, again, right at the beginning of your one-step growth curve, then a stable amount of plaque-forming units, and then if you lyse the cell at this point, you will now start to see infectious virus particles. If you don't lyse the cell now, infectious virus particles only come along later on. And what turns out, if you now look in the electron microscope, in this period of time here for adenovirus between 12 and 16 hours, you see more and more virions just building up inside the cell. Same thing is true of the bacteriophage, more and more of the virions building up inside the cell. And at 16 hours, cells start to burst and those particles come out. What that tells you is that they're infectious particles on the inside means that they don't actually have to have picked up any kind of membrane in the process. And usually, and as we'll see this later on, most enveloped viruses are picking up their membrane from the cell, usually the external part of the cell. So now if you look at an envelope virus, and again, this example here, Western equine encephalitis virus, if you look at the number of intracellular particles and the extracellular particles, they come up at exactly the same time. There's no difference between this eclipse period, which was no viruses inside or out, no virions, I should say, no infectious virions, and the latent period. The latent period and the eclipse period are exactly the same. And so what that means is you have probably something which requires that surface of the cell in order to make these infectious particles. And this is very true for enveloped viruses. Naked viruses will often have this differentiation here. So just having a difference between an eclipse period and a latent period will tell you you're likely to have an enveloped virus or a non-enveloped virus. Without even taking out a microscope. Just looking at one-step growth curves. You can get a really good idea whether this is true or not. Another thing that you can get from these one-step growth curves is what's called the burst size. Burst size is just the number of PFUs that you have at the end of your one-step growth curve divided by the number of PFUs that you have in the latent stage of the one-step growth curve. Now, why the heck does this PFU in the number of latents tell you what your virus's per cell number is? These are the same. So, PFU final versus PFU latent. So this is the latent period again, right here. It's counting, PFU per cell. Why would you see a PFU after we've gotten rid of all the infectious, all the infectious particles are gone? So where do these PFUs come from? So it's the infected cells, because if you think about the plaque assay, you're exactly right. Those infected cells are going to give you a plaque, because how do you do these assays? You get your culture, you do the dilution series, and each of those infected cells will still give you a plaque. It's not an infectious particle at that point, but each of those is still going to give you a single plaque. Does that make sense? It's a great point. Yes, no, okay, good, we're happy about this. This, by the way, is this is a quick shout out here from the Flint textbook, which should now be on reserve in the library. So if you want to take a look at that, you can go ahead and do that. So just wanted to finish up talking a little bit about math. Yes? Math, statistics. We're all happy with math and statistics. Good. So the reason for doing this has to do with the fact that to do a one-step growth curve, you have to have a high MOI. Why do you have to have a high MOI? Why do you need a multiplicity of infection of 5 to 10 if you want to have a synchronous infection? And so that gets to the Poisson distribution. And this has to do with just a statistical approach for looking at particles and how individual particles associate with each other. So you've got a large number of particles, you're mixing them together, how often are they going to interact with each other? And again, this is just a Poisson distribution. So in looking at cells and viruses, again, these are individual particles. So interactions of viruses with hosts, and I should say this is with virions and hosts, is going to be random, and it's going to be described well by a Poisson distribution. So here's the Poisson distribution down here. If I gave you a question like this on an exam, I would give you the Poisson distribution. So you don't have to remember the formula here. But the important thing here is that the proportion of now cells that have been infected by a certain number of virions here is going to be e to the minus m. M is your MOI, times m to the k, again, the number which have this particular number of particles associated with them, divided by k factorial. So everyone's clear on factorials and e to the minus m, m to the k. Good, at least we have that much. So if you look at this and just plot this out, and this is just looking at that mathematical formula. If you now look for a, excuse me, p of now a instead of k for an MOI of 2, you now look at the proportion, and this is now percentage, going from 0, and this would go all the way up to 1 here. If you have 2 times as many, say, virions relative to cells, and then you're just interested in the proportion that have a certain number that's associated with them. So for a MOI now of 2, if you look here, there's about, this is 10%, 12%, about 13%, actually a little over 13%, almost 14%, that are not infected. You get twice as many virus particles as cells. They should all be infected, right? No. According to the Poisson distribution, about 13% won't be. Quite a few will have a single particle that's associated with them. Quite a few will have 2. And then, but some will have 3, some will have 4, some will have 5, some will have 6, etc. If you do an MOI of 5, now you notice you have a pretty small number, but a not insignificant number of uninfected cells. So this is pretty good. It's about the lowest you're going to want to do in terms of your one-step growth curve to make sure that everybody's infected simultaneously. But you got some that have 1, some have 2, some have 3, some have 4, some have 5. If you've got enough virus, infectious virions, to do an MOI of 10, that's great, because you have extremely small numbers now that are uninfected, and most that are around 10, and some that are reasonably ridiculously infected. And I personally had some trouble thinking about this, and so I made up some little animations that hopefully will help think about this. So if we do an MOI of 1, so MOI of 1, 5 cute little virions, 5 poor unsuspecting cells, and we do this infection, we'll see, some of them have 1, some of them have 2, but some of them are uninfected. So MOI of 1, not so good. MOI of 2, this actually works out okay. Again, pretty random distribution. Some have 1, some have 2, some have 3, etc. But if you do an MOI of 5, you're pretty likely to have each of them that have a particular infection which has happened. Okay. Make sense? Yes? We're happy with Poisson distributions. Were I to ask you any question on this exam, so you'll all be totally happy with it? Yes?
A represents how many virus particles there are per bacteria?
Per cell, per infected cell in this particular case. And one of the things that I would do if I gave you one of these questions on an exam, one of the really nice things to do with this is figure out how many uninfected cells you have. So again, you have 0 particles that are associated with any particular cell. That would mean what's the proportion now that you have of no cells that are infected. So if you just do the calculation, again, just plug and chug here. For 0, k is 0, 0 factorial is 1. M to the 0 is going to be 1. So you're just looking at e to the minus m. So for an MOI of 1, the number of uninfected cells is 37%. Wow, yeah, one virus per cell, 37%. MOI of 2, as I showed you before, 13.5. MOI of 5, 0.7. MOI of 10, 0.005%. So ways of figuring out how you have these different numbers here. So who cares how non-infected cells? What you really want is infected cells. So the important thing to here is to think about the proportionality. So again, this is just a proportion. We're talking percentages here. So 1 is 100%. So 1 minus p of 0 is going to be the proportion that you have of infected cells. So if you're trying to do one of these one-step growth curves, and you want to make sure that as many of your cells are infected as possible, you now can figure out from your MOI what the proportion is that you have that has 0, and then 1 minus the amount that you have as 0 is everything other than 0. So an MOI of 1, 63% are uninfected. Really bad for a one-step growth curve. MOI of 2, 86%. MY of 5, 99%, MY of 10, 99.995. Okay, everybody happy with this? Everyone got their clickers? And I posted this already, so it should be pretty straightforward. So, the way we'll do this, and sorry for those of you who weren't in molecular last term, everybody gets a minute to decide by themselves. Then you can talk to your neighbor about what you answered and why. And then you get another minute to change your mind if you want to. So, we'll start our minute right now, and I'll turn on the lights, so if you want to scribble things out and try and do calculations, you can go from there.
Wouldn't have to have an extra clicker, which you haven't been able to buy one yet.
Okay, not for right now, but just scribble it down. But this today does not count, by the way. This is still just to make sure that things work. So, and this, again, clickers only start counting next week in terms of points. And there are clickers, by the way, if you need to get one just for this class, there are a couple on reserve in the library for 2-hour reserve. So you just go over there for reserves for this class. You can check one out.
No base.
You should be on A. Are you on AA? Okay. So, talk about your answers with your neighbors. Yeah, it's closed now. No, we'll let you talk, and then when you're done talking, then you get to vote again.
I don't think my answer counted for the first one, but what did you say? Yeah.
I feel like it's 13, but 5 seems more reasonable. So close.
What did you say?
You all ready to vote again? Yes? No? Whatever. Again, it doesn't count, right? So who cares? Next week, though, it will. Okay, another minute. Go. And then feel free to tell each other what you're doing here. And the thing is, I can see what differences have happened up here. So if I start jumping up and down, or five. Okay, so what do people think? Okay, we'll go over here. What I was looking for last term. Yay!
Wait, whoa,
That's pretty impressive. We didn't get this all last term. We also had about 3 times as many people, 5 times as many people last term. So hopefully this is what we'll see all the time. So yes, and this way it's also really nice because I don't even have to do a participation score. It's all just one. Which then the reason, again, part of the reason I'm doing this and they're not counting this week is so that you can make sure that your clicker is properly registered. And so I've been posting, I don't know how many of you see that you had clicker points. Anybody see that you had clicker points? Okay, good. So they've been uploaded appropriately, which are there. So if you're having trouble with registration, there's a link in D2L under announcements, and you can take a look at that there. So yeah, the correct answer is C, yes, because it's 1 minus the uninfected cells will be the infected cells, and that's your e to the minus m. So with that, just wanted to quickly finish up reviewing what we talked about today. Again, virus discovery, these were these really small things that you couldn't grow, were being filtered through these filters, could come through, still cause disease, but couldn't grow anything. A virus is alive, they're part of life, obfuscate completely. Some really cool things to do with viruses, particularly thinking about the phage therapy aspects of things. And we're all clear on plaque assays, how you make a plaque, what it means. Talk about one-step growth curves in the Poisson distribution. And before everybody gets up and runs away, there's a guy sitting in the back row who is pretending not to be here. Wave, Nacho. Here's your lecture for Friday. So he'll be talking about virus structure, probably mostly at this point, which is something he's working on as a postdoc in my lab. We're probably not going to be recording that lecture, so make sure you do lots of good notes. Or if somebody does have a recorder, bring it, and I will post at least the audio recording on YouTube so that people will have access to that. So with that, I will see you on Monday. You'll see Nacho, or Dr. Nacho as we call him in the lab, on Friday.